Welcome to Layla's Science Hub. Today, we're diving into one of the key biomolecule groups you must understand for A-Level Biology, lipids. We'll explore triglycerides, phospholipids, and cholesterol, and see how their structures link directly to their functions. Let's get into it. Lipids aren't polymers like carbohydrates or proteins, but they are still considered macromolecules. They are insoluble in water but soluble in alcohol as nonpolar due to lack of oxygen molecules. The structure of the fatty acids can vary. Saturated have no double bonds between carbon atoms, whereas unsaturated has one or more double bond. Unsaturated fatty acids create kinks in the chain, making the lipid more fluid and lowering the melting point, like those found in olive oil. Saturated fats are straight and packed tightly, so more solid at room temperature, such as butter. A triglyceride is made from one molecule of glycerol bonded to 3 fatty acid chains. This happens through condensation reactions, forming ester bonds. Each ester bond is formed between the OH group on glycerol and the COOH group of a fatty acid, releasing water. To break a triglyceride down, we use hydrolysis, so water is added to break the ester bonds. This is important during digestion when triglycerides are broken into glycerol and free fatty acids. Let's go through the key functions of lipids and how they support life. Energy source. Lipids can be broken down during respiration. Hydrolyzing the ester bonds releases glycerol and fatty acids, which are further broken down into carbon dioxide and water, releasing lots of ATP. Energy store. Because they're insoluble in water, lipids don't affect the cell's water potential. They're stored in adipose cells and release more energy than glucose, since fatty acids have lots of hydrogens and very little oxygen. Insulation. In animals like whales, adipose tissue forms blubber for heat insulation. In nerve cells, lipids act as an electrical insulator. Extra fat can even be stored for hibernation. Buoyancy. Fat is less dense than water, which helps aquatic animals stay afloat. Protection. Fat cushions delicate organs, acting as a shock absorber. And in some bacteria, peptidoglycan is protected by a lipid-rich outer coat. So lipids aren't just energy-rich molecules, they're vital for survival, from keeping warm to staying afloat. Overall, triglycerides are excellent for energy storage. They release twice as much energy as carbohydrates. And because they're hydrophobic, they don't affect water potential in cells. Now let's talk phospholipids. These are similar to triglycerides, but one fatty acid is replaced by a phosphate group. This gives phospholipids a unique structure with a hydrophilic head pointing out, the phosphate group. Hydrophobic tails point in, the fatty acids. By having hydrophobic and hydrophilic qualities, this means the phospholipid is amphipathic. This dual nature is key for their role in forming cell membranes, where they arrange into a phospholipid bilayer with heads out and tails in. The membrane is partially permeable, so only non-polar molecules like oxygen can pass through the bilayer. Cholesterol is another lipid with a different structure, a small, flat molecule made of 4 carbon rings. It sits between phospholipids in the membrane, affecting fluidity and stability. It also acts as a precursor for steroid hormones like testosterone and estrogen. The following table summarizes most of the information from the previous slides. Exam tip. Be ready to draw or label triglycerides or phospholipids. Always link structure to function. For example, branched fatty acids, more ends, quicker hydrolysis, rapid energy release. And be able to spot double bonds in unsaturated fatty acids.